A good day to everyone. This video is used to presenting the evolution of Archaea Genome project, past and present. In the late 1970s, Carl Woese and his colleagues discovered an entirely new group of organisms that lack peptidoglycan in their cell walls and having the ability to survive in extreme pH and temperature. Via DNA sequencing, this organism has vast differences in genetic makeup with usual bacteria and the eukaryotes. In the beginning, they named as Archaea bacteria, but Carl Woese decided this term was a misnomer and shortened it to Archaea. Archaea was revealed later that eukaryotic traits such as histones were also found in the genome of Archaea. Furthermore, archaeal DNA replicates using machinery more closely resembles with eukaryotes instead of bacteria. The biggest obstacle to study Archaea was the difficulty to cultivate them in vitro. After the invention of molecular techniques such as direct RNA sequencing and cultivation-independent approaches, we are able to overcome this obstacle. The first sequenced Archaea was the Methanococcus gennashi, a hyperthermophilic methanogen. It was discovered and isolated from the ocean thermal vent at Woods Hole Oceanographic Institution, WHOI. The project was done by the group Tiger, who were led by Craig Venter. The genome was sequenced via whole genome random sequencing method. First of all, the whole genome was digested into small fragments. These fragments were then inserted into the small insert plasmid library as well as the large insert lambda library to produce genome scaffolds. Sequencing from both ends was required for all the clones. Thus, assembly to form a genome map. It was reported that the Methanococcus gennashi has a circular DNA with 1.66 megabase pair and has two extra chromosomal elements. One of them is 58 kilobase pair while other is 16 kilobase pair. In the following years, cultivation-independent approaches were introduced. For instance, metagenomic, a study of the collection of genetic materials from a mixed community of organisms. Compare with conventional genome sequencing, metagenomics approach requires greater attention for sampling to ensure the sample was representative. It can be classified into sequence-based identification or function-based identification. Another modern technique is single-cell genomic. It is an emerging technique to study the individuality of cells by omics approach. The omics refers to the study of genome, proteome, and metabolome. Due to this technique was established after next-generation sequencing, it had been optimized and results in greater resolution and a better understanding of an individual cell function. This figure shows the comparison between single-cell genomic and metagenomic. The dashed line refers to false assembly contigs. There are pros and cons for single-cell genomic. This method able to display all resulting contigs originated from an individual cell DNA. However, assemblies of single-cell genomic was often fragmented or incomplete. By having these modern genome sequence methods, scientists able to study Archaea in more detailed ways. Thus, the phylogenetic tree of domain Archaea keeps expanding over the time. Until the year 2017, there are 4 superphyla, which includes Euryarchaeota, Pan, Tak, and Asgard in Archaea domain. In conclusion, advances in cultivation-independent genomics approaches has allowed us to explore the diversity of these ancient microorganisms, Archaea, better. The genomic data of different archaeal species are crucial in understanding their biochemical and ecological roles to other organisms as well as the environment, thus help us to discover the diversity of life on Earth, and hopefully, to set a step forward towards cracking the code of the origin of life.